La 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 la. Waiting, waiting, waiting. Waiting, waiting, waiting. There we are. Starting. I can see myself on the screen and I don't need to do that anymore. We've got the...
Nobody likes that.
I don't have to stare at myself. I'm going to hide that. Are we all ready for a show? I think we're ready for a show. So we are starting in 3, 2. This is TWIS. This Week in Science, episode number 682, recorded on Wednesday, August 8th, 2018. Sciencey bird funerals. But I'm Dr. Kiki. Hey, everybody. Welcome to another episode of This Week in Science. And tonight, we will fill your heads with junk, depression, and necrophilia. But first...
Disclaimer, disclaimer, disclaimer. Everyone has opinions, but not everyone forms them in a trustworthy manner. There are several things to keep in mind before having an opinion. 1, are there facts supporting my opinion? 2, other people's opinions are not facts and therefore cannot be used to support my opinion. 3, if other people have this opinion, where did they get it? 4, why do so few people or so many share this opinion? 5, why do I have or want to have this opinion? 6, is there a reason that me having this opinion is useful to somebody who is not me? And 7, understand that regardless of how well researched an opinion is, it could still be wrong. Despite the fact that most of the opinions we currently hold could possibly be wrong, there is a method of fact gathering that allows for a high success rate in opinion forming. The scientific method. Yes, there are those that are of the opinion that science is biased. And they are right, because in science, opinions don't matter at all. Not in the least. The scientific method is heavily biased against opinions and in favor of facts. So, if your opinions are based on science, they are based on a method that has filtered out incorrect opinions before you even got around to forming one of your own. And what's more, your opinion could still be wrong. Yes, but much less wrong than if you had started your opinion forming it anywhere else. And as always, the very best place to start forming opinions about anything is This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I wanna learn everything. I wanna fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I wanna know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and Kaeli. We have a wonderful show lined up for you. Another episode of This Week in Science, ready, full of science, and I am so thrilled. So thrilled. I have stories about stars, koalas, and we have an interview with soon-to-be doctor, we'll have to find out about this, Kaeli Swift, about avian death rituals. Very interesting stuff. Justin, what do you have?
Oh, that's a great question that I wasn't prepared to answer for some reason. Oh, yeah, I've got Blinded by the Blues, Vikings of Greenland, and 44 new planets, all of which I will delve into in complete detail.
Oh, that is going to be one exciting show. Planet after planet after planet. Blair, what have you brought to the Animal Corner this week?
Ooh, I brought birds on antidepressants. I brought queen bee digestive systems and some dog urine.
Oh, as you do, you know. Yeah. Got an Animal Corner. Put some dog urine on it.
Got some dog urine on it.
Yeah,
gotta claim it as my own, you know.
That's right, or whatever dogs.
Yeah,
everyone, we are going to claim this moment as our own for science. That's right. And as we jump into the show, I want to remind you all that if you have not subscribed yet, why haven't you? You can do it very easily by heading to TWIS.org, where there are links to find out how to subscribe to us on Google, iTunes, and YouTube, and also through to our podcast. Other places, if you use other outlets, just look for This Week in Science. All right. I would love to introduce our guest this evening. Kaeli Swift is a PhD candidate at the School of Environmental and Forest Sciences at the University of Washington. She has been working to finish writing her dissertation. And she studies thanatology of crows. It's very exciting stuff. Kaeli, welcome to the show.
Thank you. Thank you so much for having me. And yes, I'm in what I imagine is probably going to be ultimately the most painful part of writing and finishing my dissertation. So yes, but we're very close. And it has been many really fun and interesting years leading up to this moment of studying really interesting birds doing really interesting things.
Yeah, it's very exciting to get to that point of finally writing up and completing what is, a many years long endeavor to get the PhD degree. But it in no means is it everything. There's so much more.
Yes, there are many other ways to be a scientist than getting a PhD.
Absolutely. But let's talk about you and your work. And can you tell us and our audience, what is thanatology?
Yeah, so thanatology is the scientific study of death. And when it comes to human beings, there's lots of different ways that field can manifest. It can manifest as palliative care or forensics or medicine or all kinds of things. It's a very interdisciplinary field. And my area of research specifically is what's called comparative thanatology, which is looking at how non-human animals respond to death. So thanatology itself is this very, very big umbrella. And then I'm in this very, very narrow little corner of it where I'm just looking at non-human animal death responses.
How did you get interested in this? I mean, were you a very morbid child?
No, I'm not at all. I'm not a morbid person. I don't like scary movies. It's a very unromantic answer, unfortunately. So when I started graduate school, I had my initial idea. I knew I wanted to study crows, and I got into a lab to be able to do behavioral work with corvids, particularly crows. And my original idea actually revolved around reciprocal altruism and looking at if crows shared and what that might mean. But those kinds of studies, a lot of the really interesting, sexy stuff that people are doing right now with corvid behavior really is contingent on having a large aviary and a hand-reared captive population. So like a lot of the cool stuff that some of your listeners may have heard of when it comes to things like their perception of cheating and their ability to learn people who are unfair to them and equity, like all of those kinds of things, all of those kinds of studies happen at institutes with large captive populations.
Right.
And we didn't have the infrastructure for that at the university I'm at. We can keep crows in captivity for a temporary amount of time, but we don't have a stable population. So reciprocal altruism kind of went out the door for that reason. So then it was like, okay, so what is interesting, not expensive because I didn't have any research money, and feasible for a master's/PhD. And sort of coincident to having that conversation and thinking about what might be interesting, my advisor, John Marsliff, had already started work on doing some neurological functional imaging work on crows. And he had thought it would be sort of interesting to eventually take that to exploring how their brain responds when they see dead crows, because humans have known for a long time that crows respond really strongly to their dead. So that part of my research, just the sheer fact of crows have this really particular response or that they do something at all, is not thanks to science. We've known that for a long time. But that sort of grew this idea that would be really interesting to look at from this functional imaging standpoint. and getting some dead crow, taxidermy dead crows and putting them out is fairly inexpensive to do. And we could sort of break it up into these interesting questions that were really feasible to do within the time frame of being a graduate student. So that's why I say it's not that it was a very logistic. It was definitely informed by what interested me, but there wasn't this like long path of
going and visiting Edgar Allan Poe's grave a bunch or anything like that. Yeah.
No. I could really make up a better story for that answer because I get that question because I get it a lot.
No, I think it really gets at some of the aspects of graduate school that you have to pay attention to, that you're there for a limited time frame. You usually have a very limited budget, and you don't necessarily have unlimited faculties or places to work in. You're very limited in your scope, and there's a lot of troubleshooting and creativity in terms of how can I study the thing that I want to study with these limitations put on it. And so I personally love your story because I think it really gets into the real nitty gritty of the difficulties of science.
It's true that is such a valuable initial lesson from graduate school, is sort of appreciating, like you said, all of the logistical, financial, et cetera, challenges of doing scientific research, and sort of realizing like, oh, you have to really consider those as you choose projects. And, so there's a lot more thinking behind how science happens than just purely what is the most interesting thing for me to study right at this moment.
And I'm curious to find out what you've been finding, because also that these constrictions have forced you into studying something that I don't know if you could properly study with all the resources in the controlled environment with the lab crows. Because in the lab, death has got to be kind of an anomaly. Seeing a dead thing isn't something that's going to come about. But in the wild, there's kind of dead things everywhere. So seeing how they react to their own dead in the wild, as opposed to just something that doesn't seem to belong in our lab environment, in our controlled environment, makes it a study that possibly couldn't be done without all of those restrictions that were put on you.
Yeah, I mean, I think that's fair. This has become a really interesting, cool project that was born out of a set of limitations. But, and like you're saying, there's certain parts of this research that we have to sort of accept as not necessarily being representative of what truly happens, right? We can't account for every single thing. And one of the biggest differences in our research from a lot of the literature that's already out there on what other animals do is that we have to use unfamiliar birds for our studies. And that's a major caveat actually for the experiments that we do. But, like you said, without some of those caveats, it's really kind of impossible to even start exploring some of these questions.
Yeah, I mean, unless you were to go out and fully band and identify every single individual in a local crow population so that you would know whether or not the crows were responding to, a locally dead friend or an interloper crow.
Yeah, and then you got to wait for a crow to die and record information right away before a raccoon takes off with the body.
Which was always one of the most fun things when I was out doing research that people would ask, because they see me standing there with my binoculars, my clipboard, and they say, "What are you doing?" I'm like, "Oh, I'm doing this research study on how crows respond to dead crows. See, right up there, up ahead on the sidewalk, there's a dead crow there," and they'd be like, "How did you get so lucky that crow just died right there?" And I'd be like, "Because I'm a professional."
No, the truth is you have friends in taxidermy, isn't it?
The truth is that I have a friend who very, very generously donated a lot of his time to stuffing a lot of dead crows for me. And it's just, science is just such a great way to meet really wonderful people that like stuff like that. And so he was a former tech for me. He had volunteered on the project previously, and then on his own had decided he was going to start getting into taxidermy as a sort of a hobby and learn from our Natural History Museum at the University of Washington. And then I was like, oh, that is a skill that I would really like to take advantage of if you'd be willing to share it. And I was really lucky that he did. And so in a lot of ways, this research was really possible because of Joel and the work that he did.
So that begs the question, is your lab, is your office really just filled with taxidermied crows?
No. Because, and the reason for that is because the reason we have to go through so many crows is because they could be so aggressive with the bodies. Like if they're not coming around and screwing around with them, you can reuse the same specimen for years, right? As long as you take good care of it. But the reason we went through so many is that sometimes during a very limited time of the year, crows come down and they rip them to shreds. And so what that means is not that I have a lot of taxidermy crows around the office, but I have several boxes filled with parts.
with pieces of crows.
Oh no.
Pieces of crows. It makes getting our house ready for Halloween really easy.
Absolutely.
What this is getting to the heart of the matter though. So why are they ripping these dead fallen compatriots apart?
Or if we can go a little bit further back, I'd love to find out, what your hypotheses were going in, knowing that crows do kind of have this mobbing behavior for the fallen. What were your hypotheses going in, and then what did you learn over the course of your various studies?
Yes, let's go back. So when I started work in 2012 for my master's, we were interested in just nibbling at this question of why are they interested in their dead? And there are a lot of ways that you could potentially explain this. For example, you could say that it's an expression of grief. It is a social opportunity. So maybe they're trying to see who died and how it's going to affect the sort of goings on and social structure. It might be an information sharing opportunity, danger learning, et cetera, et cetera. And some of these things are difficult, if not impossible, to test in a sort of systematic way, grief being the biggest one that comes to mind. So my job is to sort of figure out, okay, so we have this list of potential hypotheses, which can we tackle in a systematic way? And danger learning and avoidance fit that bill and had some precedence in previous literature. So there was a study that came out right before I started graduate school looking at what are now called California scrub jays, where they showed that this behavior, which is very similar to what crows do, seems to be motivated and looks very similar to sort of their anti-predator response. So maybe it's an extension of that. So that sort of laid the groundwork for the first study that I did, which was to investigate.
Let me pause it real quick. So are they? Does that mean that they are checking the dead bodies to see maybe what might have killed them?
So that is a slightly different hypothesis that will come into play in one of the later studies. But no. So to just answer your question outright, the idea is that they're coming into an area and they're like, oh, somebody died. Everyone look out. And this place where it died is probably no good. So just be careful if you're around here in the future.
So, yeah, this is a hot spot now because somebody it didn't end well. Don't know why, but high alert.
Like when a murder has happened in a house, nobody really wants to buy that house.
Yeah, that's kind of it.
It's not
danger avoidance.
Not necessarily, but it means that there's a murderer somewhere in that neighborhood.
And honestly, as you'll see with our results, I actually had wished it was that strong of a response because there's a lot of ways in terms of management we could really utilize that. It's more like, they're not like, everybody get out of here and never come back. But they're kind of like, ooh, be careful when you're here. Like if you have to come here, just watch out.
so it's like the people who bought the house in Poltergeist. Oh, that's weird. Things are moving on their own. Well, we'll just be a little more cautious about where we put them.
Maybe leave the lights on at night, but don't move out. It's kind of like that.
No. We won't do that till the end of the movie.
So there was some precedence for this idea of sort of danger avoidance. So for the research that I did for my master's, which happened over the course of 2012, 2013, we kind of narrowed in on that and repeated it in crows, where we looked at, again, whether or not they were learning about the place. But then we also looked at if they could learn new predators who they had seen interacting with dead crows. And the easiest way for us to test that question was to look at people seeing those predators, because we knew before going into this that crows can learn individual human faces. And we just didn't know if this was one of the contexts that they were maybe learning them in. So the sort of brief summary of that experiment was I'd go out to the neighborhoods in Seattle and downtown Seattle, and I would find breeding pairs, and I would feed them for 3 days. And that sort of gave me a baseline, like on average, they come down within, I'm making these numbers up, it's been a while, I don't remember them, but say a minute. They come down a minute to get the food. And then after 3 days, we would introduce our sort of scary event. We tested several things, but one of them was a person holding a dead crow. So they would just stand there, you know, like this, holding a dead crow for 30 minutes by the food pile. And we would see kind of how they would respond. And then the person would leave, and I'd see, like, are they going to come down as soon as they're gone and get food? And then for the next 3 days, there wouldn't be a person there. And again, I would continue to feed. And so in that case, I was really trying to create this or look for a statistical difference in, for those first 3 days, they came within a minute. And for those 3 days following the scary event, it took them 20 minutes or whatever.
Longer, right.
Longer,
Was there a difference?
Mm-hmm. And then if that person who they'd seen holding the dead crow came back after a week, are they just going to be like, oh, hey, what's up? Or are they going to, you know, treat them like a predator and mob and scold? And in both cases, we found that the answer was yes. So they didn't just never come back to the food, but they were clearly, it took them longer. And you could just sort of see that sort of antsy when they were in there trying to get those Cheetos, they'd be like, I really want it's just like this very clear anxiousness. And then when they'd see that person come back after a week, they would dive bomb and scold and mob.
Really? And that person is not holding a dead crow the second time.
And that person is not holding.
Just recognizing. Wow.
And that would go on for 6 weeks. So we'd bring that person out once a week for up to 6 weeks. And in 40% of birds, they went all 6 weeks. So
hopefully they don't communicate this language or this event over generations. Because that poor person's like, yeah, everywhere I go, crows all hate me everywhere. Apparently they can talk to each other, and they pass this information down to generation to generation. You can't go outside anymore. Just get dive bombed by crows the whole time. Horrible.
Yeah, no, because they can. That's true. They do that.
Oh, no.
Yeah. I couldn't tell if you were making a joke or because you didn't actually know that, but they can.
This person is now going to be tormented by crows, and it turns out it's true. Oh no.
It is true, and that was one of the reasons. Wasn't the main reason, but it was actually one of the reasons that our volunteers did not go out there holding the dead crows themselves, but they wore these masks to conceal their identity and make it so that they could roam the streets of Seattle, protected from
not being mobbed by crows. Yeah.
That was some good foresight.
So that was the first study where we were like, okay, great. They learn about danger. That's a nice, clear narrative. And then one day, a little bit after we published that study, we were trying to capture some footage of how the crows respond. And so we had put a dead crow out, and like I just described with the experiment, in that first one, somebody was always holding it, right? Or it was like near red-tailed hawk or some of the things I didn't talk about. But just suffice it to say, it was always very close to a predator that would prevent crows from ever wanting to get that close to it. And so for this, moment that we had, we were just, again, trying to collect some footage of how the birds respond. So we just sort of put one of our taxidermy crows out in the grass, and this was in the beginning of March, kind of it was in the beginning of April. And the crows came in just like they do. I was like, all right, recognize this, I know what's happening here. But instead of the usual explosion of scolding and then this gathering, like we had been seeing with this first experiment, the crow got down on the ground and it sort of sauntered up to our dead crow, and it wasn't scolding yet. And I was like, okay, I've seen this before. Like sometimes they really like to go and like really give it the eye, and before they freak out. But this crow didn't. It just kept walking up to it, and then it dropped its wings and erected its tail, which is their pre-copulatory display, and it got on top of the bird, and it mated with it. And then another bird came into a crow. Whenever I say bird, just to be clear, I mean crow. Another crow came into the tree and started scolding, and then the first one was like, ah, okay, and got off, and then it started scolding, and then it was like the normal mob. So then we were like,
what just happened?
Yeah, was that guy just really confused, or
like, what's happening? No. So then that sent us on sort of this new trajectory of like, oh, there's this whole new question that we didn't even realize we needed to be exploring that we should explore now. And so that kind of laid the groundwork for the next series of experiments where I would just put dead crows out, and there's sort of different studies, but they all have the same format. So I find a breeding pair, I put a crow body out, and in one of the experiments, we were looking at if, how frequently and what the nature of these tactile interactions would be. In another experiment, I was looking at whether or not they pay attention to the age of the crow, if it was a dead adult or a dead juvenile. And then the very last set of experiments that we did were these functional imaging things like I talked about at the top of the show. So that's kind of everything in a nutshell. It's a lot that I just threw at you, but that's kind of all of the pieces.
And so with the question of the odd behavior, not just, it's probably not just necrophilia, but just a kind of inappropriate behavior with a dead conspecific, right? Do you have any hypotheses about why that's happening? I mean, are these sociopathic birds? Does Crow Society have sociopaths? What is going on?
So our initial hypothesis for this behavior was, okay, so depending on the nature of the response and how frequently it's happening, those are sort of two things we need to pay attention to. We might explain it that it's motivated by information acquisition, which we touched on earlier. So maybe they're coming down and they're touching these crows as a way to investigate, like, does it have obvious wounds from hawks or cats? Or no, maybe they're doing some kind of investigation like that, or they're trying to ID the individual. Maybe it's a misuse of an adaptive response. So during the breeding season, they become much more aggressive, much more territorial. Maybe they're so amped to attack that they don't take the time to pay attention to dead or alive. They're just on it, ready to do their thing. And then they don't realize until it's too late, so to speak, that it's dead potentially. And then they're like, oh, well, that was a waste of time.
Yeah, and you would think too, like the response from the other crow in that scenario would normally be to move quickly away. So if they weren't interested, so there is this, so there's not a holding up and sitting back and sort of contemplating so much as it's a lot of aggressive trying to sit next to the other bird, trying to sit on the same branch really quick, and then maybe get a little closer and it flies away. And so then that's like, circle around, try it again. And so this one, it's like, oh my gosh, this one didn't move away. This is my shot. I gotta, and then too late to really do the analysis.
Yeah, so maybe it was a problem like that. Maybe it's cannibalism, maybe crows are trying to eat each other a lot more frequently than we realize. So those are some of our main hypotheses going into that particular study, looking at the tactile interactions. And what we found was that first and foremost, this behavior did not happen very frequently. It only happened about a quarter of the time. And so what that suggests to us is that it's probably not motivated by information acquisition, for example, because if that was the case, we would expect it to happen more often and more sort of uniformly across the breeding season. And so the fact that we found that it was relatively rare is maybe not quite the right word, but it's certainly not characteristic of how they respond to dead crows. That seems to fit with this narrative that we had started with, which is that it's dangerous to engage with a dead body, right? So it exposes you to disease transmission potentially, parasites, dangerous scavengers. So the fact that most birds don't do this fits with this idea that generally dead crows are really about avoiding danger rather than engaging with it. But then that still leaves this quarter of birds. So what is going on with them? Do they have some sort of deviant behavior? And so with that group, we found that sexual interactions and these really aggressive, ripping them to shreds and making you have to have all of these taxidermied crows, again, was limited, and it was very seasonally limited. So even within the breeding season, that kind of behavior pretty much stops by June.
So during probably the most competitive portion of the breeding season.
Exactly.
Yeah.
But interestingly, so one of our other ideas was, so these dead crows, I wish I had one, but I don't have any in my house right now.
I wish I had a dead crow. That's how many.
The way that Joel had initially prepared them was always sort of with the wings kind of out from the body so that the tail was exposed. If I stand up, the wings are kind of like out here, right? With the tail exposed. And that can sort of superficially look like how a female presents when she's ready to copulate. So we wondered, maybe the position of the stimulus is releasing this behavior. So we did a follow-up study where we presented crows with our traditionally prepared birds with the wings out. We showed them dead crows, but where the wings were tucked really tight into the body so that it didn't mimic that posturing. And then we also presented them upright, lifelike crows. So they had glass eyes, they just looked like they were standing in the grass. And with that sort of side experiment, we wanted to look at, one, if the position of our dead crow was releasing the sexual behavior, and two, if maybe they didn't guess that these birds were dead, right? That they were just on it, attacking it, and if that was the case, we would expect that how they treated these upright birds would look very similar to how they treated these dead birds. And what we found from that experiment was, no, this behavior does not seem to be, the sexual behavior does not seem to be released by the posture. They tried to copulate with the birds with the really tight wings and our lifelike upright birds just as often. And the second thing we learned is, despite the fact that they would try and copulate with these upright birds, outside of that, their responses were actually quite different and much more consistent with how we would expect them to treat territory intruders, living territory intruders, than with these dead birds. So that sort of helped us identify that this isn't just a really simple stimulus release response, right? They're not just having this reflexive response to something about our stimulus. And this behavior doesn't seem to be driven by just a pure, they're not, they don't understand that it's dead and they're just sort of mistakenly doing these behaviors. So ultimately, where that leads us is, it's probably something about the hormonal changes that these birds go through at this particular time of year. And for a minority of birds, that change downregulates their ability to effectively process a conflicting stimulus. And what that means is basically, by conflicting, we saw that these stimuli could evoke fear, right, in this scolding, mobbing response. It could evoke aggression, and it could evoke sex. So that's the way in which it's conflicting. Most individuals can look at that and kind of work through it and be like, ultimately, fear is the most appropriate response. So I'm going to stay in my tree and scream. But a minority of birds are just like, so many hormones. I'm just going to do all the things. And I kind of describe it as either like that feeling of wanting to call your ex at 3 a.m, where your prefrontal cortex is like, this is a bad idea, dude. Don't do this. But it's tired and kind of worn down, and so your reptilian brain just takes over and you do that. Or the other analogy is like middle schoolers, right? I mean, middle schoolers are in many ways like very intelligent creatures, but they're just at the mercy of some crazy hormones.
Yeah.
And that's really, messing with their ability to always make the right choice. So.
Yeah, and the one that came to my mind first was like, we've all had that friend that was dating somebody that we could all tell this was not a good situation. Like, this is bad news. How come you don't see this? You're otherwise a rational, reasonable person. And yet they're like, oh no, he or she is great. We have a wonderful time. But the fact that they just got out of jail for the 3rd time since you've been dating and it's 2 months, like, that should be screaming things. But yeah. Okay. So these, so there are.
Yeah, we have hormonally charged crows in the spring engaging in questionable behaviors The one
But
part of the
definitely not
study that I
majority.
yeah, but the exactly. I'm really fascinated. You mentioned briefly the brain scanning, and back when I was in grad school working with zebra finches and very small migratory birds, there was no brain scanning technology for birds. I had to slice up many bird brains, and so I'd love to hear what the advances in technology have allowed you to do with crows.
So it's really interesting because our functional imaging study is not actually really a reflection of an advancement in technology. It uses a technology that's been around for a long time. What it really reflects is the hard work of a small group of people who figured out how to put a crow in a PET scanner, how to like actually just change the machine. But the technology behind it, and it was used, they're using it in this novel way, but it is interesting because it's actually kind of an old school approach where essentially what we do for these functional imaging studies is we collect crows from the wild. We have a small aviary where we can keep them temporarily. So we just let them get acclimated to captivity and kind of wind down a little bit. And then when it's time to image a crow, we bring it down to our imaging room and let it stay there overnight, get used to the sounds of the room, and it's in a smaller covered cage. And then when we're ready to go, we take the bird out of the cage and we inject it with a modified glucose molecule that has a radioactive tracer attached to it. And so it's called FDG. And the basic principle of this is that when you are thinking about something or processing or doing something, whatever part of your brain is the most engaged in that activity is going to be using the most glucose. So the idea behind this study is once those birds have that chemical floating around in their brain, we can show them different things. We could show them a dead crow or a red-tailed hawk or the face of a familiar person. And whatever part of their brain is like working to process that information is going to be concentrating that molecule.
Mm-hmm.
And the reason I called this a modified glucose molecule is it's been modified in such a way that it has a much longer half-life than a normal glucose molecule. So it sticks around in the brain for about 40 minutes after, well, about an hour and a half total. But once we've shown them all of these things, we have about 40 minutes where that molecule is still stuck in whatever part of their brain was using it. And we can anesthetize the crow and put it in a PET scanner. And that scanner is going to detect that radioactive tracer, the flag that was attached on that molecule. And when we sort of average that across all the birds that saw a particular type of stimulus, and we compare that to our control group. And that allows us to visualize what parts of the brain were active when they saw a dead crow, for example. And then the best part of this is once the birds come out of the scanner, they have to stay in the radiology room for about 24 hours to let all of the radioactive material process out of their body. And I should add for your listeners, this is exactly the same kind of process that you would have as a human being if you were having a tumor PET scan, for example. And then after that, they're free. They go home. So, it's pretty wild to be able to do brain studies on an animal that not only doesn't require euthanasia, but where they actually get to like go be, wild birds again after it.
That's
it's
awesome.
a very cool technique. Yeah.
Oh, I became vegetarian for years after after my grad school experience, and I just kept waiting for this kind of technology to be used in this way. The resolution, are you able to what kind of resolution in the brain are you able to get? Are you able, like, when we image the brain and we look for changes in activation, it's like, oh, fear response, the amygdala is lighting up. And we've gotten it so that depending on what kind of like fMRI or whatever we're using, we can get some pretty fine scale information of what part of the amygdala even is becoming activated. So in the bird brain, what are you seeing? Because I'm in the crow's brain, it's like a walnut. It's a bit smaller.
So it's funny. You would probably very much appreciate this, having done zebra brain work, because the thing about these studies is that John learned very early when he first started doing this is you cannot take these PET scanning images to folks who do the kind of research that you did with these slices and be like, what am I looking at here? Because they're like,
I don't know.
I have no idea. This is so different looking. And the avian brain is really different than the mammalian brain.
Yep.
Partly because the sort of wrinkles on the mammal brain that are so iconic, birds don't actually have that. Their brain is completely flat, which is like a whole other cool thing that we don't have time to talk about, but that we could. We're basically like, crows do so many things that we do and have figured out how to do that with so much less area, like volume in their brain. It's wild. But anyways, there's also, unlike the human brain, there's no obvious sort of landmarks, is what our radiologist calls it. It's very flat. And so we can pinpoint to general region, but it's not quite as high of a resolution, at least my understanding of it is not, that we can sort of go down to say the different sub parts of the amygdala, for example, or the NCL. But basically what we have to do is we have an atlas, a brain atlas, where it's, you know, a slice by slice image of the jungle crow brain, which is a species in Asia. And we match up the coordinates from our PET scanning images to the coordinates on this atlas. And then we're like, okay, this looks good, this area. So, but it's very different than the process of doing fMRIs with people, where you're like, oop, I know this, I know exactly what this looks like. So yeah, it is different in that way.
Yeah, and there haven't been as many ablation studies in the crow either. I mean, you know, I know we've gone into the zebra finch brain and, you know, taken out little regions of the brain to see what happens. You know, the hippocampus has been ablated, and we've learned how the hippocampus in the bird brain is involved in learning and memory and behavior and all that.
Hand raised in the
And
back.
Yeah, and we've done
ablation.
Ablation. It's when you use
Ablation?
acid or nail scissors or something to just kind of excise the region, get rid of it.
Oh, okay.
Ablate. And in humans, we know we have, like, you know, lots of neuropsychiatric studies from disorders where people have undergone strokes or had disease where it's damaged parts of the brain, and then they have behavioral changes. And so we've been able to pinpoint that with much more exactitude. And so it's amazing that we're starting to do it in the bird brain for these more complex behaviors, like the social behaviors. This is fascinating to me.
Yes. Yeah. And it's very cool. But like you said, it is a little bit more esoteric because, you know, we're not going in and just sort of pounding out these particular parts and saying, what happens now? And, you know, so the drawback of that is that it's a little bit more esoteric, but the advantage is that I think at least with what I know about crows, that's not a study I'd want to do. Giving crows brain damage on purpose.
Ooh, they'll never forgive you for that one.
Well, unless you damage the right region and they can't recognize you, or they forget you. Yeah.
Right. Yeah. Unless they damage their hippocampus, and then they'd be like, oh.
Yeah.
Exactly. The crow version of HM. It would be, yes, an interesting one. So you're moving on with your dissertation. I know we've been talking with you for quite some time, and I appreciate all of the time with you.
45 minutes.
I know, it's so great.
You just can't stop with crows. They're so interesting. There's just so many little tangents you can go on, and yeah, I mean, I could stay here all night, but I know you got other fun things to talk about.
For you, where are you going next? I mean, I know you're finishing your dissertation, so what is the next step?
The next step is that I'm going to be switching gears kind of totally and not at all. I'm going to be staying with corvids, fortunately, but I'm going to be going up to Denali National Park and actually looking at foraging strategies and behaviors in gray jays up there.
Oh, cool.
Yeah. So I'm really excited to, work in nature and not in urban neighborhoods anymore, because I'm excited about having to think more about bears than human beings in my field type. That is a transition that I am welcoming at this stage. But it would also be fun to sort of switch gears into another corvid, and then also a sort of a totally different aspect of their behavior than I have been able to look at previously. So I'm very excited.
Are you going to be working with Russ Balda or somebody out of his lab?
Not directly, no. So this project is a collaboration between actually my current lab at the University of Washington and National Park Service. So I'm still going to be working out of John Marshall's lab technically.
Fantastic. Okay, gray jays. For everyone if you don't know gray jays by the name gray jay,
yeah,
and also camp robbers. They're the friendly little gray birds that come in and want to eat all of your food, and they look so cute when they do it when you're out camping.
Yeah, and they are now called Canada jays, and I still
Canada
haven't
jays.
disabused myself of the gray jay name. So, but Canada jays is what I'm doing.
Awesome. And for people who want to, you've got a wonderful blog for people who aren't aware. It's Corvid Research blog, and we will put the link to that on our website. Are you going to keep up with your blogging and letting people know about the work that you're doing?
I am. So yeah, I'll probably be doing regular sort of Denali diaries. And one of the things that for my graduate work that I just didn't get around to doing because I didn't have all of the tools and I was a little wrapped up was really sort of taking people along for the ride of what fieldwork is like. But that's something for my postdoc that I'm really excited to do, and sort of these daily updates and letting people see kind of how fieldwork happens and what it looks like. So yeah, my blog, corvidresearch.blog, Instagram at Corvid Research, and Twitter at Corvid Research. So if you just remember that part, it's very easy to find me everywhere.
Corvid Research, Corvid Research, Corvid Research. I love it. And there was one last thing I'd noticed. Running through your blog, a comment about, or I think you were answering somebody's questions, a comment about the sounds that crows make. And when I was working in a bird lab with scrub jays and magpies, I found that they didn't always squawk and screech at you, that sometimes there were these nice little whistles and little sounds that they make. And have you heard that with crows? And do they seem to make these friendly sounds to each other?
So corvids are just amazing vocalists, and all of the different groups have an incredible repertoire. Ravens might actually be my favorite, but crows have the caw, right? Everyone knows the crow, like caw, caw, and they can sort of vary that if they're more or less stressed out. But they have a lot of what you're describing, which are these sort of quieter sounds. They have a water drop sound, it sounds like dripping water. They kind of make these like these like wow wow calls are what we call them. They can sort of whisper and whistle. They'll knock, kind of like knocking on wood, or kind of rattle like a kid's rattle. And I mean, crow vocalizations are probably the area of corvids that have had some of the most like human energy with the least results. Because we still know so little about these calls because they are so context dependent, and so why they use some of these really quiet sounds is still a mystery, because sometimes they do it when they're alone, sometimes they do it when they're with their partner. So yeah, but if you spend enough time watching crows and you do so in an area that's not completely overwhelmed with anthropogenic noise, you get to hear a lot of these really, really cool sounds that they make. And I can't tell you how many bird walks I've been with students who are just getting familiar with birding and learning birds, and they'll be like, oh, what's that new sound? What cool bird is that? And I'm like, oh, it's a crow. And they're like, what? I didn't know they made that sound.
Exactly.
So,
yeah.
Yeah.
I love it. I love that they have those. I love those little sounds. It's like they're the intimate sounds of the corvids.
Yes, very much so. And yeah, I think those are the little details that remind people that there is this whole other intimate side to these birds' lives that, I think when they're just yelling outside of your bedroom window at 6 a.m, you forget about, and you can sort of be kind of irritated at them. But crows have these amazing social bonds, these very long-term social bonds, and there's this intimacy about corvid sort of life and relationships that is really worth exploring and getting into, because it's really fun to watch and learn from.
That's wonderful. I love how much you love corvids and the work that you're doing. Thank you so much for sharing with us today. It's just been a joy.
Yeah, thank you so much for having me.
been wonderful. So everyone, don't forget corvidresearch.blog, Corvid Research on Instagram and Twitter. Yes?
Yes.
All the places if you want to find out more. And we are going to go into our break right now. We're going to take a short break. Once again, Kaeli Swift, thank you so much. And you're almost done in the last rush of your PhD years. Almost no longer a candidate, soon to be full PhD.
Soon, we hope.
Yeah. Good luck. Congratulations. All of the above. Yes. And we hope that we'll get to talk with you eventually about more of the corvids at some
I'd
point.
love that.
That'd be great.
Can
All
this
right.
be our go-to? Like, could you be our go-to corvid person?
Please?
Like,
yes, can I?
yeah, like I think that's it because we need that because they come up enough. So we need the bigger updates once in a while.
Beep, beep, beep. We'll get the corvid phone.
Yes.
Please, I would love that so much. Yes, I have my little intro sound just squawking. Oh, yes, I can.
Yeah.
Awesome. Okay, we will talk about this. We'll definitely be in touch. All right. Have a wonderful night. Enjoy the rest of your evening. And thank you so much, everyone out there. We will be back in just a few moments with more This Week in Science because we have some science stories. I've got koalas and viruses and stuff, and there's more. So stay tuned.
Can't explain things you've heard with more than intuition. A lot of reason shows the way to go. New conclusion. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see. The answers lie somewhere within this scatter plot. Our first assumptions were correct. Please prove the rest. It's hot. The methods of hypothesis and patience are the only things I need. Put on a pair of goggles and go looking for the things I couldn't see.
All right, everybody. Thank you so much for listening to us tonight. And I would love to remind you as we take our break that you can help keep TWIS going week after week after week. We've been here 682 weeks now and more. That's just like an estimate, kind of. Not really. But yeah, we've been around a while. So you want to help keep us here longer? You can do that by subscribing to This Week in Science. If you have not subscribed to This Week in Science, an easy way to do that is to simply go to TWIS.org, our website, where you can click the big orange subscribe button. That button will show you links to YouTube, iTunes, or Google Play, the three top places where you can subscribe to us. In addition, you can just search for This Week in Science on just about any podcast directory. We're on Spotify now and all over the place. Subscribe, listen, follow. That's right. That will help keep us going, and tell people to do the same. If they want to know what you're listening to, tell them. Don't keep us a secret. Please tell somebody about TWIS this week. Get somebody to subscribe. That would be amazing. If everyone who listens to this podcast got one person to subscribe to the podcast, oh my goodness, how amazing would that be for everybody involved? Our community would grow like wildfire. In addition, you can help us out by donating. You can click on the yellow donate button on the sidebar. That will allow you to use PayPal to donate one-time amount of your choosing. It's a very easy, simple way to do it. You can also donate by clicking on any of the recent episodes, scrolling all the way through the show notes. The show notes, have you ever been there at our website, looked at the show notes, all of our links? We got links to all these stories so you can investigate them further. Go down the rabbit hole. At the bottom of those pages, there are pink donation buttons that will allow you to donate in a recurring fashion. And finally, If you would like to help out in a donating way, you can do so in another recurring way by clicking on the Patreon link in our header bar, which will take you to our Patreon community where you can choose the level of your support. And the great level to start at is the $10 a month level. It's just charged once per month, $10 a month. And we will thank you on the air. That's right. It's really about $1.50, $2 per episode ish. If we're going for, or maybe $2.50 per episode. $10 a month. We'll thank you on the air by name if you like. But there are other levels of support. Click on that Become a Patron button. Also, if you like stuff and you want to wear TWIS goodies around, you can click on our Zazzle store link. A Zazzle store is where you can find all sorts of fun stuff, t-shirts, mugs, pillows with logos, and also with original art from past Blair's Animal Corner calendars. So many fun ways to support TWIS. Subscribe, donate, or buy our merchandise. All of these ways you can definitely help keep TWIS going week after week, month after month. But it's all about you because we really could not do this without you. Thank you for all of your support.
Can't explain things you've heard.
And we're back with more This Week in Science.
We are back with more This Week in Science. And right now it is time for This Week in What Has Science Done for Me Lately? Well, Susan Barnum wrote in to our website. She left a comment over at twist.org. She said, "Hi, here is my What Has Science Done for Me Lately entry. I love dogs, and I have a pack of dogs living with me and my kids. One of the wonderful things about science are all of the amazing types of medicine we have developed for our furry friends. There are also vaccinations. We've also, pest control and more. Also, the fact that dogs are one species and so different from one another in size, color, furriness, etc., always blows my mind. I enjoy thinking about how they've been with us for so long, as scientists discovered by looking at the genetic clock, and how we've most likely influenced each other's lives. Taking my dogs to the vet means that I get to spend even more quality time with my canines. Thanks for your awesome show. I love what each of you brings and how you have unique voices and perspectives." Thank you, Susan. Thanks so much for writing in. And I think this was a fun. Your perspective was fun in thinking about all those wonderful aspects of variety of dogs and how science has brought that variety to us, either through breeding and genetic variation. I mean, people are trying to clone their dogs now. There's all sorts of sciencey stuff with canines.
This is the one thing that I have learned over the years that I kind of wish wasn't true. Because I still like the idea of the huge packs of Chihuahuas,
Right.
hunting in mass on the plains to put down. Like that image that there was this primitive, free, wild Chihuahua. It's gone.
Yeah.
The opinion I had has been.
I do like how she mentions that we've influenced each other, because I think that's such an important thing to remember, that as much as, we may think that we've tamed wolves and turned them into dogs, they have tamed us into being their pack mates and feeding them every day and picking up their poop. So it's a pretty good deal for them.
It's a fantastic deal. Our furry four-footed friends. Everyone, remember, we need you to write in and let us know exactly what science has done for you lately. Has it, I don't know, interested you in penguins more often? I don't know. Is it what?
Penguins.
Penguins?
Somewhere there's a podcast
Shrews?
of dogs talking about the domestication of man.
Yeah, for sure.
We'll see. Somebody write in, though. You can write me a note at Facebook if you use the Facebook. Go to This Week in Science, our page, and send me a message there. You can also email me, Kirsten, KIRSTEN, at thisweekinscience.com. Kirsten at thisweekinscience.com, or our Facebook page. Leave a message. Tell us what science has done for you lately. We want to keep doing this segment of the show, and we can't without you. It might just be silence, or we will have to start doing something else. But if you like this segment of the show, send something in, please. Should we talk about science now?
Yes, bring the science.
Yes. So we've been looking at our own sun for quite some time. And we've known for a very long time about these wonderful, this phenomena in our sun known as a coronal mass ejection. And they usually occur after solar flares. So there's a burst of light from the sun, a solar flare, and then whoosh, the corona of the sun ejects a whole bunch of energy, these very high energy things. And it's like, whoa, this is our star. This is our sun being active and, bursting with its electromagnetic dynamo fields that's happening and making all this stuff happen. Yeah, lots of exciting stuff happening. But if we were to look at other stars, would we see the same activity? Can we, with our telescopes, see coronal mass ejections on other stars? Well, researchers...
No, I'll be very disappointed.
No, that big lead up, right? No. Nope. Well, we can't.
Yeah.
Yes. Yes, we can. Yes, we can. Astronomer Costanza Aguirolfi of the University of Palermo in Italy and colleagues found evidence of a star's outburst, this giant star called HR 9024 that exists about 450 light years from Earth. It is a giant star. It's 3 times larger than the sun and about 10 times wider. So it's big. It's something we can see. And they used a new method for analyzing data taken from the Chandra X-ray Observatory. And this was just reported at the Cool Stars 20 meeting on August 2nd. The team detected material moving on a loop of plasma that extended from the star's surface during the flare. And they detected this by measuring X-ray Doppler shift. So the change in the wavelengths of X-rays as the material, Doppler shift, when something gets closer and passes, and then, something's passed, you have the audible Doppler shift. Well, the Doppler shift works at all levels of the electromagnetic spectrum. And so this X-ray, the X-rays can be determined to be moving toward or away from the star on a loop of plasma. Another astrophysicist from Harvard Smithsonian Center for Astrophysics, Julian Alvarado Gomez, said people have looked for this for a long time, and this is the first time this has been seen. The ejection contained about 1 billion trillion grams of material. And so this is based on extrapolations, based on what the sun's coronal mass ejections look like and how much larger this particular star is. But the kinetic energy that they measured based on the X-rays, again, was lower than was expected for what they think the size or the amount of material was.
Interesting.
So I have a question. So this star's big, like real big.
It's bigger than ours. Yeah, that's right.
So you said it was 3 times larger, but 10 times wider.
Yes.
I don't understand that.
Puffy. It's puffier.
So
massive. 3 times more massive.
Oh, okay. So the mass is 3 times, but it is 10 times larger because there's air in there. It's puffy.
Yeah, it's not as dense. So the mass is spread out over a larger area. It's a puffier star.
That was really hurting my brain.
It's more of a cotton ball sun than a bowling ball
Cotton ball
sun.
sun. Okay. Mhm. Good.
Yeah, so this is pretty exciting, and this may be the beginning of looking for more of these coronal mass ejection emissions on other stars in other systems to see how other stars stack up energetically to our own. How big are the mass ejections? How do solar flares on other stars take place versus our own? And we can test hypotheses for star dynamics as we look at this kind of stuff. So this is it's exciting news.
So far we think our star acts pretty much like other stars, but it's tiny.
It's a little kind. It's not tiny. It's just not. It's average. We get an
Oh,
average.
it's okay. It's average.
Get an average star on the small side of average. Yeah.
Yeah.
I try and think, positively.
It's good. If it was any bigger, we'd be dead. So I'm okay with it.
I'm totally okay with it also. Speaking of dying, sometimes when you get norovirus, you feel like you want to die. And norovirus does unfortunately kill in some cases. Norovirus, the wonderful virus that leads to vomiting and diarrhea and all sorts of gastrointestinal discomfort. And it's this virus. It's amazing. Usually families get it. You hear about norovirus on cruise ships. You hear about it just happening, and it just spreads very quickly. It's incredibly contagious. How is it so contagious? Well, for the longest time, we have really considered viruses, when you think of being infected by a virus, you think a virus, that there's one little viral particle that got in and somehow managed to get through all your defenses, and this one little viral particle somehow infected you and made you that sick. There's something wrong with that idea. The recent evidence over the last decade or so is starting to build up. In 2015, it was discovered that polioviruses can cluster together when they invade a cell, and so it makes them stronger as a group. Well, it turns out also that that norovirus and rotavirus, another virus that leads to gastrointestinal distress, that these viruses of the GI tract, they also band together. They can be found where, with strength in numbers, combined together, bound by a membrane, a vesicle, a membraned vesicle. So they're basically, when the viruses take over the cellular machinery and get the cell to make new viral particles, the cell doesn't just spit out viral particles. It makes them, binds them in a membrane, and then spits out a little packet.
Like a cell of its own?
Like a cell of its own, but filled with infectious particles. And looking at the study that was just published in Cell Host and Microbe, it found that these vesicle-bound virus packets are much more infectious than single viral particles on their own. So really what's happening is, like, when it's you can imagine one little virus being on your finger, and you lick your finger and it goes through your gastrointestinal tract, and it comes in contact with a cell, and the cell's like, go away. You're just one little virus. But a vesicle containing a lot of viral particles, that's going to make it. The vesicle actually, the membrane protects the viruses and shuttles them much more safely.
Viking longship.
Yes.
So you know what this is reminding me of, and I apologize, but this is all I can think about, is how octopuses mate and how squids mate, and they have their sperm packet. So they make a sperm packet, and they send it down their specialized tentacle, and then they stick it up into the mantle of the female. They're like, there you go, there's the packet.
Have the packet. There you go.
And it's kind of, so it's like how nature repeats itself in these ways when it figures out a good way to do a thing. And it's this idea that this packet gives you a better chance than broadcast spawning and hoping that just one sperm will make its way over there. Here's this little sperm grenade, if you will. There you go. And this is the same. It's a virus. It's a little virus power pack.
That's right. Yep, they've got the buddy system going on. Yeah, inside the power pack.
So what I thought initially I'm thinking of is who created this idea? Where did this come from? It was this initially a cell's attempt to sequester away the virus. I know what I'll do. I'll take all the virus that's here and I'll wrap it up in this little bundle, and therefore I'll be safe from it because it'll all be stuck in there. Is this something that the cells do to other sort of invaders or toxins? Is this something akin to taking on a heavy metal and sort of wrapping it and sequestering it in that way? And then with the virus, it's just become this, oh, really? This is actually going to work for me. Now we can move around together, and we can, when we do find someplace else to land, we'll all be right here together, having built up a good little colony. It's an interesting. You went silent, Kiki.
Oh, we can't hear you.
I had muted myself very briefly, but I'm not muted any longer. Cells do produce vesicles for little communication packets. They communicate using vesicles that can bud from one cell and then connect to another cell. They're
how yeast breed.
Yeah. So this vesicular production with the viral particles that are already kind of hacking the cellular machinery for their reproduction, it's not surprising that the cell would just go, oh, I'm making these little protein bits, or in this case, it's, viral DNA, but let's just wrap it up and send it out to go. Yeah. So anyway, this could potentially, understanding this could help us produce better treatments for things like norovirus, knowing that these vesicles are potentially protecting them from the immune system and from other treatments and therapies that we currently have. Maybe we can produce things that will target these vesicle membranes and weaken them so that we can actually get rid of these viral particles once and for all.
Is there a neuron creation a vascular system?
Synapse, yes. So vesicles containing neurotransmitters bud from the presynaptic bouton.
So this is really, we're talking from yeast to synapses. We're talking about a core architecture within cellular life that has been maintained throughout the existence of cellular life, really. Yeah. And so, and then yeast have been along from this ride long enough that whether it's just a happy niche that they're falling into, or something that they themselves have been able to co-opt.
It's a fascinating question, and people are going to continue researching that story for a long time to come. And finally, continuing on the viral theme, I'm going to jump into also koalas.
Viral koalas?
Viral koalas.
Koalas are viral creatures, aren't they? Don't they all have
well, they all have herpes, right? No, yes.
No, chlamydia.
Chlamydia. There we go. We've got chlamydia. Koala chlamydia. Anyway, researchers at the University of Queensland are studying a virus that infects koalas. And they're hoping that by studying this virus, it can demonstrate how viruses have historically altered the DNA of humans and various other species by becoming quote unquote junk DNA. One of the lead researchers, virologist Professor Paul Young, he says retroviruses insert their genome into the host's chromosome, from where they make more copies of themselves. Some can also infect what are known as germline cells, which alters the host's genetic code and that of all of their descendants. Retrovirus insertions in humans date back more than 5 million years, so we don't really know when the first interactions really took place. And so what they're hoping is that by looking at this new interaction in koalas and that it'll help us understand more about ourselves. They found about 10 years ago a wild koala population being invaded by a retrovirus. And while it's not great for the koala, it's this opportunity for study. And so they're taking a look at why and how it happens. This retrovirus is relatively young, less than 50,000 years old. So that's young on the viral scale of things. And so it's not yet fixed in a certain location in the koala's genome. And so scientists, I don't know if they're going to study it for the next 50,000 years or so to see how long it takes for it to become fixed, but they're interested in this quote unquote early engagement between a retrovirus and its host.
Yeah.
Well, they got to cure that koala chlamydia situation in order to study them for the next 50,000 years, or there won't be any. So, got to get on that.
Yeah. Get on it, scientists.
Yeah.
That's right. Justin, what do you have?
I've been blinded by the blues from time to time over the years, but not like this. Recently published research in the Journal of Scientific Reports finds blue lights from digital devices acting upon molecules in the eye's retina to disastrous effect. Optical chemistry research at the University of Toledo says this effect can lead to age-related macular degeneration, which, as it happens to be, is the leading cause of blindness in the United States. Quotey voice here of Dr. Ajith Karun Arathne, assistant professor, University of Toledo, Department of Chemistry and Biochemistry. We are being exposed to blue light continuously, and the eye's cornea and lens cannot block or reflect it. It's no secret that blue light harms our vision by damaging our eye's retina. Our experiments explain how this happens, and we hope this leads to therapies that slow macular degeneration, such as perhaps a new kind of eye drop. The backstory on macular degeneration is not a pretty sight. It's an incurable eye disease that results in significant vision loss, starting on average in a person's 50s or 60s. Although if this can be put on by digital devices, I assume that age is going to start dropping quickly. So what it does is it creates the death of photoreceptor cells in the retina. Those cells need molecules called retinol to sense light and trigger a cascade of signaling to the brain. You need a continuous supply of retinol molecules if you want to see, says Karunathan. I'm messing up this name, sorry. Photoreceptors are useless without retinol, which is produced in the eye. The lab found that blue light's exposure causes retinol to trigger reactions that generate poisonous chemical molecules in the photoreceptor eye. This is Kasun Ratnayake, a Ph.D. student working in this lab. It's toxic. If you shine a blue light on retinol, the retinol kills photoreceptor cells as the signaling molecule on the membrane dissolves. Photoreceptor cells do not regenerate in the eye. When they're dead for good. So they introduced retinol molecules to other types of cells and to other types of cells in the body, such as cancer cells, heart, and neurons. When exposed to blue light, these cell types died as a result of the combination with retinol. Blue light alone or retinol without blue light had no effect on the cells. So that's an interesting cross experiment there. No activity was sparked with green or yellow or red light. The retinol-generated toxicity by blue light is universal. It can kill any type of cell. Wow.
Great. So let's kill bacteria with it. But I mean, this gives a new meaning to blinded by the light.
Yeah.
Blinded by the light.
or blues legend Blind Lemon. Researchers found that a molecule called alpha tocopherol, a vitamin E derivative and a natural antioxidant in the eye and body, stops the cells from dying. However, as a person ages or the immune system is suppressed, people lose the ability to fight against the attack by retinol and blue light, which is so there is something keeping younger eyes healthy. It's not until you get it, but still, I'm thinking about all of the places, and they go through. This is Dr. John Payton, who's a visiting assistant professor there. He says, if you look at the amount of light coming from your cell phone, it's not great, but it seems tolerable. Some cell phone companies are now adding blue light filters to their screens, which he thinks is a good idea. They're also looking at or advising people to wear sunglasses that can filter both UV and blue light when they go outside, because the sun is a source of this blue light as well. Avoid looking at your cell phones or tablets in the dark, because I guess I would assume that the night mode is emitting more blue light than the day mode of our portable electronic devices.
I think actually on my phone, the night mode is more yellow and less blue.
You're colorblind, Blair.
No.
It is.
Justin, this is real.
No.
But it's more the contrast. It's that when you're surrounded by darkness and you have the bright light, it does more harm to your eyes in general.
Mm. Yeah. Every year, more than 2 million new cases of age-related macular degeneration are reported in the United States, says Karun. I'm getting worse at the name. I think I got it right the first time. And then
the main researcher says.
Main researcher on this. By learning more about the mechanism of blindness and the search for a method to intercept toxic reactions caused by the combination of retinol and blue light, we hope to find a way to protect the vision of children growing up in a high-tech world. Absolutely, because this is, and then there's also, if you look around, there's like the little sleep modes on the computer or the other electronic devices. They all have this little blue light or there's always these little LED lights everywhere now. It seems like we could probably do without them now. We know if a thing is on or not, whether or not it's working. Do we need all the little diagrams?
So
a
few months ago at work, my monitor, which I'm sure was from the 1990s, went bad and died. And they bought me and everyone else in the office these new monitors because there was a nice sale. And they are wide. They're super wide. They're like 2 feet wide, probably, which is great because I can work in 3 windows at the same time and my productivity has probably gone up. But I noticed really quickly that I started getting eye fatigue very fast. And when I went to mess with the settings for the first time, brightness was like all the way up. The color balance was very high on the blue spectrum. I had to move it towards yellow so it was more soothing on my eyes. And I noticed that I was getting way less eye fatigue and eye strain as soon as I changed that. But so these big fancy LED monitors are set up to have this extreme bright blue light because it makes colors look more vivid. It makes everything look better and clearer and crisper. But in the end, we weren't really supposed to see bright white light. It's like when you get your first CFL bulb and you've been using an incandescent. You're like, wow, everything's really bright, but it's a lot lighter in here than it was before.
Yeah, there's also a thing that's like a trend. It's at least a product you can buy, which is LED blue light therapy. Supposedly good for the skin. Wouldn't recommend it. And I think we've even covered in the past that there's a melatonin reaction that they thought could lead to cancer in mice by like too much light exposure. So
yeah, but everything causes cancer in mice.
So everything does cause cancer. But to go out of your way to put blue light on your face because somebody somewhere thinks this is a good skin therapy, maybe reconsider light therapies. It's good to have your body in the darkness.
reconsider. Do you want to do another story before we move on to the animal corner? Do you have another one, Justin?
I do have yeah, I do have one more. Yeah. Oh my gosh. Yeah, this is the Icelandic sagas that tell of Eric the Red, who apparently committed murder and had to leave and fled to the only place that there was for him to go, which was Greenland, and thereby establishing its first Norse settlement. There was already other people there in some small number. So the colony took root. By the mid-12th century, there were 2 major settlements with a population of thousands. By the end of the 15th century, a mere 300 years later, if my math is correct, they weren't there anymore. There was abandoned ruins and some old crumbling churches, and they were gone. So
churches,
past their
very small rocks.
Yeah, a bunch of rocks. So there's been this why did they leave? And there was all these theories, and it was, well, maybe the one of them is their farming techniques. They stuck to the same farming techniques, and it kept failing. So eventually, they decided we can't grow anything here. There's also been talk. There was some climatic changes that took place. One of the theories was the trading in walrus tusks. So part of that, though, has had this asterisk next to it. We know that the Greenlanders traded in ivory from walrus tusk, but how big was this, of a trading thing, right? Because we know that one of the walrus tusks aren't just available in Greenland. You don't have to go all the way to Greenland to find walrus. They can come from Russia or Norway. They're there's the eastern ranges of open waters have walrus available. So it seemed like they were going way out of their way if this was the main commodity by which they were living on, for them to be able to fashion some sort of lifestyle from this. So some researchers went and looked at this. This is researchers from the University of Cambridge and Oslo. They studied ancient DNA from what they're calling offcuts of tusks and skulls. These are shavings or pieces of ivory found at former ivory workshops across Europe. And they traced the origin of the animals used in medieval ivory trade. And in doing so, they actually discovered that there was an evolutionary split in the walrus. That was one walrus that was everything east of Greenland. And because they'd been trapped behind ice during some ice age, the walruses between Greenland and Canada were different. They were genetically different, identifiably different. And what they found was the walrus supply likely came from Greenland as a near monopoly, because they had the eastern walrus, but then at some point completely switched to western walrus for hundreds of years as the Norsemen had taken up residence there. So, and then all of a sudden, the Norsemen are gone from Greenland. And it also times out to a bunch of events. One is there was a suddenly African ivory had made its way into Europe. Plus, there was this Black Death thing that wrecked the economy. And and there was another mini ice age. There was a whole bunch of things that sort of happened at once. One of the other possibilities is that the walruses just decided to go hang out somewhere else, because they've been known to do this. If there's too many predators at a particular coastal area, the walruses will just leave and be gone for decades at a time. They'll just go find other places. So
Overhunting, if it was that much of a monopoly, overhunting could have been a possibility.
It could have been just overhunting and population decline amongst the walruses that did this too. But it was enough of these factors that happened at the same time frame that they likely overlapped. And so we may be able to say that the farming techniques, the Norsemen, were just fine as long as they were still subsidized by the one. And then again, it also tells us, never put all of your tusks in one basket. Okay. If you have an economy based on one commodity, if anything happens to that one commodity, your entire economy predictably will collapse.
All right. Good advice based on science.
Yeah.
And yes, history. Oh, I love it. Is it time? Oh, wait, we're not done.
It's time.
Is it time? It's time for Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's animal corner.
What you got, Blair?
I'm depressed.
Oh, no.
Yeah, I'm depressed because there's these sewage treatment plants that are leaking antidepressants.
Wait, then, oh.
How's that for a catch-22? A study from the University of York in the UK wanted to study birds at sewage treatment plants and around where these treatment plants have been found to be seeping different pharmaceuticals, including Prozac, other antidepressants. So after measuring these sewage treatment plants, they found that there was a significant amount in these areas. And then they wanted to see what this might do to some of the animals in the area. And in this case, it was starlings. They wanted to see if there was a behavior change when starlings were around these sewage treatment plants where there were pharmaceuticals. So what they did was they saw that there was these sewage treatment plants that were leaching these chemicals these pharmaceuticals, and that these pharmaceuticals were going into the soil was ending up in grubs and bugs, and then it was ending up in the starlings. So what they found was not that the starlings were more happy or outgoing or spent less time in the nest, as you might expect. Actually, they saw some negative impacts, which included that males responded differently to females that were exposed to Prozac. So in two ways. One, there was increased male aggression towards females who received dilute doses of Prozac in the lab. But two, males sang half as often and half as long to females that were on low doses of Prozac as those that were untreated. So the untreated females, twice as often, twice as long with male song, which means that's directly impacting how these animals might reproduce based on Prozac. So this is not a hormonal thing where there's birth control and there's the feminization of frogs, which we've talked about, or it's messing with sexual preference in that way. No, this is something that's not supposed to be related to that at all, that is having an impact on a community based on this. And we don't know why. We have no idea why this impact is being felt and why Prozac would make females less appealing to the males. But we do know that it's happening. So
wow. So the females are on these antidepressants, this Prozac, and they're less attractive. The males are not courting them and are in fact aggressive toward them. So it's like it's affecting. Wow, it's what signals would it be causing the females to give off that would reduce their attractiveness, and also make them, if a male is attacking them, that means that they see them as like a potential aggressor. Also,
right.
like a potential
they're seeing them as a territorial
competitor.
They're seeing them as a territorial threat because they're already happy, which means they must already be happy.
Right. So that's the question here. That's really
looking for a mate.
That's I think the next step here is to see what the behavior change in females is, if it is measurable based on this. So I would expect that to actually come first, but it seems like this study, they stumbled across this change in male behavior first. So now the question is, are females acting more like males when they're on the Prozac? Are females not accepting advances when they're on the Prozac? Are females acting less interested? What's going on here? Why is this Prozac causing this? Or is there some sort of hormonal impact that is a chemical signal that's being impacted by these pharmaceuticals? So we don't know, but we do know so far that there is an impact here. And I was talking to somebody about this earlier. They went, how is it that we still don't have proper filtration systems on sewage plants for pharmaceuticals? And it's hard. It's hard
really
to
hard.
figure that out. Not to mention, once we figured it out, it would be extremely expensive to apply to every sewage treatment plant in the world.
Well, if you think about how antidepressants are supposed to act, they are supposed to act on our brain, which is notoriously hard to get stuff into. So they, bind up to receptors, but they have to get past the blood-brain barrier, get into the brain. So they're tiny.
You're talking about molecular filters.
Yeah,
absolutely.
Really tiny, tiny, tiny little particles, not, big clumpy proteins. This is, much harder to clean out of the water.
So mental health is very important. I'm not saying that antidepressants are bad, but I am saying that in our sewage treatment planning and in our updates of these systems, I think there's some real research that could be done here, and some finding and some invention that could help save a lot we species in the long run if we can figure this out.
But
I got it. I know what it is.
What?
We go back to outhouses.
That could do it.
That would do it. Yes.
Yeah, absolutely.
Yes, no more sewage treatment plants. We'll just
everybody's got an outhouse now.
And moving from female birds on antidepressants, I now want to move to queen bees and their gut microbiome. So something that if you're a bee, you really want to be a queen bee. Justin, do you know why that is? Besides just, it's good to be the queen. Why is it good to be the queen? Do you know?
No. Do they live longer?
Yes, they live much longer. Worker bees die after a few weeks. Queens can live years. And they can be genetically identical.
Right.
So how is this happening? Well, a new piece of research from University of Arizona wanted to look past the genetics to the microbiome. And this study suggests a connection between bacteria in the bee guts, the food they eat, and the physiological differences related to aging, stress, and longevity. So once again, it's all about the microbiome. So they were able to identify the species and amounts of bacteria living in honeybee intestines, and they could compare and contrast as these animals aged, looking at workers versus queens. In humans, of course, we have our probiotic bacteria like Bifidobacterium and Lactobacillus, and we think about them as being associated with health, longevity. And then we have our not so good bacteria, the proteobacteria, often associated with unhealthy imbalances that can lead to disease and death. There are similar trends in bees. As workers age, their gut microbiome shift away from a dominance of beneficial probiotic microbes, and their intestines are taken over by bacteria associated with poor health and shorter life expectancy in bees. Queens, on the other hand, they support and manage a refined, efficient microbiome, retaining signatures of youth.
So, okay, so the worker bees, they go out, they're out in the world, they're exposed to more. I get this. But the handing off of being the queen bee from one queen bee to another. Now I got to know how that happens. I hand this scepter over to you. And by scepter, I mean I'm going to expel some of my gut bacteria that you're going to have to consume in order to be the queen. Like what happens? How do they?
What do worker bees eat? Do you know? Pollen. They eat pollen.
Pollen.
That's how we get honey.
But what do queens eat?
They eat, what is it, royal jelly? No.
Royal jelly.
Yeah.
Yes. So
Special.
this study points specifically at royal jelly, which they think enhances the growth of queen-specific gut microbes, sets the queen on a trajectory to longer life, and that this being fed exclusively the royal jelly creates specialized glands. These glands help keep this microbiome aloft. And they say you can think of royal jelly as a type of superfood equivalent to breast milk, supporting beneficial bacteria and containing antimicrobial peptides. So the researchers from this study think that honeybees could make an excellent model for human microbiome research because of striking similarities, but also because the bee microbiome is much smaller and easier to examine on a large scale. While the human microbiome comprises thousands and the honeybee has only about a dozen bacterial species, it's much more manageable. And so they think that this might actually be able to extrapolate, and they'll be able to see from this how you can maintain a healthy microbiome, what in our microbiome is allowing us to age. So this is a very exciting study for me for many reasons.
Just eat all that royal jelly.
Yeah.
So the queen bee is generally larger. Yes.
Yes.
Mm-hmm.
So is that now not? So then is it just an affect of longer life that it continues to grow? And would a worker bee, if for some reason they had the lifespan? Because I've always wondered, like, how is it that one bee looks different than all the rest if they've got the same genetic makeup? Or is there a genetic selection being taking place by this gut microbe that's allowing for a different type of growth? Or is it a hormonal expression or something?
Yeah, that's a great question. I think it's most likely something being turned on in the genetic code that's allowing them to grow larger, but I don't know for sure.
But if one's weeks and one's years, couldn't that just be around longer? You kept growing. You just got
but but it's possibly the bacteria in the gut allow for better fat mobilization, more fat mobilization. You have bigger fat stores, and then they've got special storage molecules for energy and for the youth and vigor. There are special molecules and proteins that are activated, but I'm sure it is a give and take between the bacteria and the physiology of that queen bee.
Yeah. So the bee gut might hold the key to eternal life. My very last silly story that I'll just throw in at the end here is all about dog urine. So those of us that have ever walked a dog might have noticed that if it is a male dog, he might lift up his leg to relieve himself on an object. And I always kind of joke around when I see, especially dogs at dog parks, they're just running around going, "That's mine. No, actually, it's mine. No, it's mine. No, I told you it's mine." So the marking of things is besides just marking your territory, it's also kind of leaving your signature behind. So dogs can, by sniffing pee that another dog left behind, they can learn gender, age, fertility, health elements, all sorts of things. But one thing that, as far as we know, you cannot tell by the sniff of the pee is the dog's size, which makes sense because, as we were just talking about mere minutes ago, dogs are all the same genus and species, even though they have this wide variety of all sorts of things. So this study from Cornell University, this very important study, wanted to look at taking dogs out for walks and observing and videotaping how they peed. What they wanted to see was the angle of the trajectory of the urine and the angle that the leg was raised to when they peed.
Are they trying to pee higher than the other dogs?
So, what they discovered was that little dogs lift their legs at a higher angle than bigger dogs, causing their pee to be applied higher on targeted objects than otherwise.
Yeah.
Which means these little dogs are trying to trick bigger dogs into thinking they're bigger than they actually are. Which, if you've ever met a toy breed of any sort, checks out.
Yeah, makes sense.
Yeah. So now you know.
Now you know.
I've
Napoleon
never,
complex is strong in the Chihuahua.
Yes.
Yes.
I've never thought of it as this is mine. I've always thought of it as like a check-in you might do on
Oh,
Facebook
I was here.
at a restaurant. Yeah, like, oh, Spot checked in that he was at this fire hydrant earlier today. Oh yeah, I'm gonna right back at him.
Well, let me tell you, I was at a dog park last weekend and I saw a Chihuahua pee on a bush, and then a Bichon came and peed on the bush, and then the same Chihuahua came back and peed on the bush again, and then the Bichon came over and peed on the bush. It went on for about 10 minutes. I was like, how are they not running on empty? But that was definitely them saying like, oh, this is mine. No, it's mine. No, it's mine. No, it's mine.
Wow.
So it depends. It can mean either or both.
But you always need to look at the crook of the leg.
Yeah, that's right. The angle of the trajectory.
The angle of that trajectory.
Yeah.
My last story for the evening is an interesting one. I was brought to it thanks to Reddit. Chronic forced cocaine exercise. Chronic forced exercise inhibits stress-induced reinstatement of cocaine-conditioned place preference.
That's a lot of words for the end of the show. I'm not sure.
It's a lot of words for the title of a paper. There's a lot of stuff in there. Really, what it gets down to is that exercise. This was a study in rats. They got rats addicted to cocaine and used to taking cocaine in a very specific place. So they were tested for cocaine conditioned place preference. And then they were split into two groups: sedentary rats who were not allowed to exercise, or exercise rats who got 6 weeks of 1 hour daily treadmill running 5 days per week. Each of the groups were tested for extinction behavior, getting rid of their behavior of their cocaine place preference. And then they were stressed out and they wanted to see after this 6-week intervention period of exercise or no exercise, they stressed them out, and then they checked to see which ones went back to cocaine. And they found out that rats that exercised were less likely to go back to their cocaine habit.
Does that mean the kind of the receptors in the brain, you could say, that are receiving the endorphins from exercise are the same ones receiving whatever reward from the cocaine?
Yes, so that that there is a reward pathway involved, that it gets in effect short-circuited by exercise, and that exercise could potentially be a modulation of these pathways that could lead to behavioral moderation. Yeah. Anyway, yeah. The idea is that stress, when people get stressed out, the possibility that they will use drugs increases. And so maybe if people exercised more, their use of drugs would be less.
There you go.
Just thought. Justin, do you have any more stories for the end?
I've got just one more left. I've got one more story left. This is an international team of astronomers pulled data from NASA's Kepler and the ESA Space Telescopes, as well as ground-based telescopes in the U.S. John Livingston was the lead author of the study, who was a graduate student at the University of Tokyo. And their combined resources led to the confirmed existence of 44 new exoplanets. Boom, 44 new planets. Now, Sit back as I describe each one in detail.
Yeah.
Well, well, not quite, but some of it's pretty interesting. 4 of the planets out of the 44 orbit in less than 24 hours around their sun. So that's a year a day, right? Or the 16 of them were about the same size as Earth. And the smallest was about the size of Venus, which they think is cool because it's about the smallest size planet that they think they can even detect. So, yep. As of today versus yesterday, so to speak, there are 44 more planets in the universe, that which we did not know existed before.
Nice.
More places where we could potentially send our great grandchildren.
Or viruses.
Or viruses, yeah.
Or an avatar.
Or vesicles.
Ooh, we can
Or a
create a little
robot.
packet of viruses and send it into space so that it can, or microbes, so that they may possibly. We should just start pansperming the universe just in case there's nothing.
That's right. You know, the idea that maybe panspermia led to us here on Earth. Who cares about that? We'll just take care of it for us for the future. Panspermia, the universe with the humans.
Good idea.
That's
Whether
right.
It's like
whether
manifest
right
destiny.
Manifest destiny. Here we go.
That's right. There we go. Oh, you guys, thank you for another wonderful show. Thank you for inspiring me to dream larger, that this universe is even bigger. 44 planets bigger now.
Yeah.
That's right. And I will look at dogs a little differently.
Good.
Thanks to you.
Crows and dogs a little differently.
And thanks to our guest, Kaeli Swift. Yeah, I will look at the crows with a little keener eye. Thanks to our guest. Again, our guest was wonderful, talking about crow behavior with the death of crows. If you're interested in crow death behavior, then you can check out her stuff, corvidresearch.blog, Corvid Research online. All right. Thank you, everyone, for listening to the show. Thank you for watching the show. Thank you for being a part of our chat rooms. If you are in our chat rooms, thank you for being there. Fada, thank you for helping out with monitoring the chat room over on YouTube. Thank you for help with the show notes on the YouTube description and for helping with social media. Identity4, thank you for recording the audio for our show so that we can have a podcast every week. And I would love to thank our Patreon sponsors. Thank you to Mark, Tyrone Fong, Keith Corsell, E.O. Byron Lee, Kevin Parachan, Mark Hessenflow, Matt Sutter, Aaron Luthen, Flying Out, Christopher Rappin, Brendan Minish, Greg Briggs, Robert Gary S., Marjorie, Rudy Garcia, Kurt Larson, Steve Lesseman, Ben Rothig, Sean Lamb, Greg Riley, Jim Drapeau, Lisa Slazewski, Christopher Dreyer, Brian Carrington, Jason Olds, John McKee, Paul, Rick Ramus, Artyom, Richard Porter, Shara Chavez, Ulysses Adkins, Jacqueline Boyster, Ashish Pants, Jim Seabright, Noah Zemke, Christoph Zuknurek, Dave Neighbor, Kevin Reardon, Noodles, Aiden Jeff, Richard, Brian Condren, Richard, Eric, Knapp, Kyle Washington, Time Jumper 319, Bob Calder, Bill K, Jason Roberts, Matthew Litwin, Jack, Mark Mazzaros, John Ratnaswamy, Craig Landon, Ed Dyer, Tony Steele, Alex Wilson, Steve DeBell, Andy Groh, Joshua Fury, Charlene Henry, Harrison Prather, Ken Hayes, Richard Onimus, G. Burton Lattimore, and Paul Disney. Thank you for all of your support on Patreon. And if you're interested in finding out more about how you can support us through Patreon, you can visit TWIS.org, look for the Patreon link, or you can go to patreon.com/thisweekinscience. Remember, it's easy to help TWIS out. Sometimes it just takes telling people about us and what we do. On next week's show, we will be back again, 8 p.m. Pacific time on Wednesday evening at twis.org/live or our YouTube channel at This Week in Science. If you can't make it, don't worry, because we will have our past episodes all saved there for you for posterity.
Thank you for enjoying the show. TWIS is also available as a podcast. Just Google This Week in Science in your iTunes directory, or if you have a mobile type device, you can look for TWIS, the number 4 Droid app in the Android marketplace, or simply This Week in Science in anything Apple marketplacey.
For more information on anything you've heard here today, show notes are available on our website. That's at www.twis.org, where you can also make comments and start conversations with the hosts or other listeners.
Or you can contact us directly. Email Kirsten at thisweekinscience.com, Justin at twistminion@gmail.com, or Blair at blairbaz@twis.org. Just be sure to put TWIS, TWIS, somewhere in the subject line, or your email will be spam filtered into oblivion. You can also hit us up on the Twitter, where we are at TWIS Science, at Dr. Kiki, at Jacksonfly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to you in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
Ka, ka. Oh, wait. No, it's all in your head.
This week in science. This week in science. This week in science. This week in science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. This week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got one disclaimer and it shouldn't be news. That what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just get understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. And This Week in Science is coming your way. So everybody listen to everything we say. And if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 I've got a laundry list of items I want to address. From stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This Week in Science is coming your way. You better just listen to what we say, and if you learned anything from the words that we said, then please just remember it's all in your head. Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. 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 This Week in 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 Science.
And we're at the end of the show. Once again, the after show. And Kai is still awake. Why are you making ugly faces? Yeah, because you're tired, huh?
I'm saying, yeah.
Why are you saying wah? You're making ugly faces at the camera.
End of show.
Yeah, it's the end. Oh, you're saying wah because it's the end of the show.
And what's that? And who's that?
What? Who's what?
Oh,
What's
that
what?
was you.
Yeah, I put the microphone in the headphone in
No.
your ear so you can
No,
hear
the thing
Blair.
back there on the broadcast.
Oh,
Go back at the
no, that's not the broadcast. That's just a website.
No, just go back a bit on those photos.
Oh, yeah. Not everybody can see it there.
What's that?
Me.
What
Just
was that?
looking at it. Yeah. It was just
What
a
was that,
pic. It was
though?
just a picture on my website, love. It was.
Did you let it your video?
Tired child.
I get
This
it.
is what happens when the father is out of town for work. I don't have anyone to put him to bed at bedtime, so
Oh my
he comes down
goodness.
here with me and doesn't go to sleep because what were you watching? YouTube videos. And you stayed up watching them.
Yeah, what YouTube videos, Kai?
Honestly, I have no idea.
You don't even know what you were watching.
I was pressing random videos.
Oh, yeah.
What?
Probably all Minecraft.
Oh, Minecraft.
Not all.
Not all. No, okay.
When do you go back to school, Kai? Do you know?
Month?
Next month?
Yeah, it's like
Somewhere in the middle of next month.
it's like through 2.5 weeks actually.
Oh wow. I
Pretty
have
soon. Too soon.
some of my teen volunteers start tomorrow.
Whoa, that's early. They must have
Yeah.
gotten out earlier than
Yeah,
Kai
I
did.
guess. I don't know. Yeah, are you excited to go back to school, Kai?
Yep.
See your friends? You're going to be in second grade, is that right?
Yup.
My goodness.
Don't yank on that, please. It'll make the audio funny. Please don't touch it. Yeah. Well, if you yank on it, it can mess the connection up. And we don't want that. Don't mess up the connection. Let's see. Ed from Connecticut. I'm going to be traveling with Kai to NYC. Kai, do you like flying? Yeah.
What are you
Yeah.
doing in New York?
Visiting?
Oh, just for funsies?
Yeah, for funsies. And Marshall bought the tickets. And so I am going, and I'm going to be in New York over next Wednesday evening, which means I don't know how I'm going to do the show.
That's right. We'll do it. We'll take care of it. Right, Justin?
Yeah, of course.
Yeah.
Yeah.
It'll be
Okay.
fine.
You could maybe do it in New York.
Yeah,
Yeah.
but I would have to start the show at 11 o'clock at night in New York.
And you
And
won't be
in
that time adjusted.
not just that, it would be in the single room hotel room
Yeah, where
that
everyone's
we're
sleeping.
staying in, where everybody else would be sleeping.
They
Not
have
to
lobbies
mention that
that
hotel
nobody else will be
Wi-Fi
using.
is
Be
a
in the
nightmare.
corner at the desk in the room going, "This is This Week in Science."
No. The hotel lobby, which nobody will be in at 11 o'clock on a Wednesday. That'd be
Should
fine.
I'll do the show from the lobby.
Yeah.
Not.
Great.
What could go wrong?
It's
Nothing.
me being super effusive
Yeah.
in front of my camera, in front of my computer. Oh my God. People would be,
Would love it. You'd
no.
gather a crowd. I'd be
I'd
all
be
over
the weirdo.
No.
I love it. I love seeing one
Kiki,
side of a
just
conversation.
ship us out there on
Yeah.
short notice and we'll do the show live.
Oh, yeah.
We
No,
love
I
the
can't go anywhere on a
NYC.
Wednesday. I can't go anywhere.
Anyway, no, it'll be fine. Just I can never remember how to set up the event. So if you can still do that, I'll do everything else.
You go lie down for a second. Go lie down for a second. No more iPad. Yeah, I will set it up.
Yeah.
I will
That's
set it
all
up.
I
if you
I
do
can hit the
everything
button.
else. You can hit the button. I can hit broadcast.
Yeah.
Yeah, I got to figure out how I want to go have a nice time in New York City, but I'm like, hey, yeah, I like my work thing also.
He knows better. It's been 680 something episodes. He's like, you do what on Wednesdays?
The YouTube chat room wants to know
Really?
if the pet is still, but that might be a sensitive subject to talk about.
Oh, the one that ate the toxic plant. She's okay, right?
A cat?
Your
No.
cat.
Stella.
No, not the cat.
Oh.
Cat is fine.
The craw.
The crawdad
Oh.
disappeared.
No.
What?
Disappeared.
I told you.
So like
One
we
left.
saw it the last night, and then the next day we thought it was just growing a new shell, but then a few days after we checked everywhere, and he was nowhere. No.
Did the
So-
cat eat it?
No,
I don't
so
think so. I mean,
told
it was
this.
nowhere.
That's possible. I told this story a while ago of a friend who had some crawdads over to his backyard to visit, and one of them escaped and ended up at the neighbor's yard. And the neighbor thought it was an alien insect and called
Mhm.
animal control because they'd
Mhm.
never seen a crawdad
Mhm.
before.
That makes
And were
sense.
terrified by this thing. But they're crafty. That one had to get out, get away from everybody, find its way over, under, through a fence to get into the other yard. Like it was pretty resourceful.
I keep wondering where we're gonna find the crawdad. If he's gonna be like living somewhere.
In your pillowcase. Ah!
Yeah. Or but
Way to
then
give a kid nightmare juice, Blair.
That's all right. Kai's got much worse things on YouTube waiting for him, I'm sure.
yeah, but then so it could be a cat because no, not anywhere in the aquarium, all gone. And then we got Kai some fish. How many fish did you get?
8,
How many fish are there now?
7.
What happened to the fish?
One disappeared.
A fish disappeared.
Cats.
Yeah.
I think,
No, it's
but
most likely-
it's been
most
now it's
likely
been
filter.
now it's been a
Or
couple
other
of
fish.
weeks and there's still 7 fish. It's been fine.
That's really good.
Maybe
Did you
the-
give this fish tank a good stabilization time before you introduced fish to it?
yes.
Okay, because I didn't. So I went from like a dozen to 3 before not realizing I was supposed to have this like week-long stabilization event take place before I introduced fish to it. I did it all wrong.
Did you install a black hole to the fish tank?
Why would I do that?
How
That
about
would
a
kill all the
wormhole?
fish.
Not a wormhole?
That would probably
fish
bring
like
it onto
wormholes.
land and then it would die.
Oh, true.
Yeah.
Very true.
Yeah.
Okay. Just checking. Just checking.
Yeah.
Hmm.
What do you think? A wormhole? Do you think the fish and the crawdad are in outer space?
They would die.
They would die. Yeah.
I mean, they'd still
They're
be
not
there. They'd just be dead.
They'd just be dead, floating in space, frozen, dead fish.
Happy
fishy-
and sad.
happy and sad.
I'm happy
Yeah.
and sad.
You're happy and
Duality.
sad.
Yeah.
It's funny, but then it's not.
Yeah. That's true. I think your crawdad is living it up somewhere.
Yeah, the fact that it disappeared, I think he escaped and he went somewhere.
He's like under the fridge or something.
Yeah. But now Kai wants to get more crawdads.
Maybe it bit through. Maybe it crawled into the not the freezer, freeze. No, maybe it went into the cabinet. That thing.
Oh yeah, you think it went in the cabinet?
Yeah, and then it went into a bag, a really cold bag with this.
Oh, interesting. I think you should get, instead of a crawdad for your aquarium, I think you should get a cleaner shrimp.
We already have a snail.
You have a snail? Yeah. Get a shrimp.
would be cool.
Or harlequin shrimp. I love those. That's too fancy, probably. Those are like very expensive, I think.
And we just have a freshwater aquarium.
Yeah,
without
there's
a heater.
freshwater
Right now it doesn't
shrimps.
have a heater. It would have to be freshwater. So right now we just have some little freshwater fishies.
Let's see. Freshwater aquarium shrimp. Oh, cherry crystal red shrimp. Ooh, cleaner shrimp. They do have cleaner shrimp.
Cool.
And ghost shrimp. Those are really cool. Ghost shrimp. You could also get some sea monkeys.
But are turtles freshwater?
Turtles are freshwater usually.
Yay, but turtles.
Yeah, but you'd have to get some sort of like terrestrial element for them because they can't stay in the water forever.
Don't they also like aren't they
out.
like heavy? What is it? Salmonella?
They can shed salmonella, but as long as you're not touching the turtles and then licking your fingers. No, you can touch them. Just don't lick your fingers after you touch them. Wash your hands. You're fine.
Salmonella poisoning from pets?
So Justin, this is how I explain it to zoo guests all the time because we can't let anyone under 5 touch reptiles because they shed salmonella, right? And they're like, "Oh my god, that's terrible!" And I'm like, "No, think about it like this: human guts have a normal microbiome. They have bacteria that just live inside, and it doesn't hurt us, but it might hurt another animal, right? It's exactly like that. It's part of the reptile's natural microbiome, and so it's just part of being a reptile is having salmonella just on your body sometimes.
Yes.
And so
like it's part of being a 5-year-old, putting your hands inside your mouth all the time.
Exactly. So if you're over 5, then we follow up a pet right away with some Purell or a hand wash and you're fine.
Ooh, let's talk about the Purell story from this last week.
There was a Purell story? I missed it.
Well, the story was that strains of bacteria in hospitals, and it's not like it's not a big deal yet, but there are bacteria that are potentially becoming resistant to alcohol gels.
Wow. So,
but this isn't cause for super concern yet. Wash
your
hands. Still just wash your hands.
This does, because I am, if I have a cut or a scrape or something of this nature, my instinct isn't to put an antiseptic Purell or a antibiotic soap or anything like this. I just take some rubbing alcohol, some ethanol, and I pour it on the cut. And I'm like, I'm good. You're not going to get, resistance to alcohol, all right, from these bugs. I'm just killing everything. I've done a good job. Tell me they build a resistance to alcohol, we will all die.
That is true.
That is bad.
I don't use it in my everyday life really, except for I use it if I have been handling animals and I am not near a sink. Then I use it because I don't want to accidentally like touch my face or something like that, right? Without thinking about it. But I also, every time I go to the doctor, I use it a few times because I am so paranoid about whatever creepy crawlies might be in the hospital or in the doctor's office where I am, that on the way out, I always grab a little pump of hand sanitizer. But that's pretty much the only time I use it. Oh, and camping. If you're like, it just whenever I'm not near a sink where you'd really want to wash your hands and you can't, I would use it.
Yeah. So for the most part, they aren't completely resistant yet, but they did find an increase that bacteria gathered from 2 hospitals in Australia between 1997 and 2015 appeared to gradually get better at surviving the alcohol used in hand sanitizers. And it seemed to be in step with the hospital's gradually increasing use of alcohol-based sanitizers within that same time period. Yeah.
Yikes. But like, what's the alternative is the problem.
Yeah. Hand sanitizers often contain 70% or more isopropyl or isopropanol or ethanol, which usually annihilates bacteria. The alcohols shred the microbes' outer membranes that cause them to leak out. So high concentrations of alcohol are quickly fatal to bacteria. Yeah, there needs to be more research done, but it's still fine. Your hand sanitizers are cool still.
So that's why I use a 70% solution.
More of it. Way more.
Yeah.
Like, it's like like a giant, like 2 buckets of it.
2 buckets. Just stick your hands in buckets of hand sanitizer.
No, like get 2 buckets of the materials, like 2 buckets of each material, and mix them together.
Yeah.
That'll make a really strong one. It'll just destroy everything.
That's good.
Maybe it'd be so
Mix the
strong to destroy all the
isopropanol and the ethanol together.
That's actually maybe a little bad idea.
Mhm.
If I did that, I would probably destroy all the bacteria in the world. I would destroy all the bacteria.
Oh, that would not be good. We need the bacteria.
Yeah.
Yeah, that's actually a problem.
Yeah.
Yeah. You're
Wait,
right.
maybe it isn't. Wait, is it?
It is. It is. So
Yeah.
the method of antibiotics that are out there, the microbes are becoming resistant to them, so we can't kill them. But we need microbes as well in our digestive system. So the alcohol treatment that does kill them, that they don't build a resistance to, and we can target, so that we only kill the ones that are outside on surfaces of things. Those are good. But we don't want to mess with our own internal microbiota to get rid of those, because we need those to digest everything, to make and to keep our bodies healthy and happy.
But just don't pour it in your mouth.
No.
Yeah,
Yeah,
that wouldn't be good.
Don't drink
always a
alcohol.
That would be really yucky.
Never drink alcohol. That is a good lesson that we should always be focused on. Yes.
Is that
No.
filled
No.
with alcohol?
No, that was not. Why would you think? It's just because it's a red cup wouldn't make that for a. Whoa, what's that? Past my bedtime? Okay. Say good night, Blair.
Good night, Blair. Say good night, Justin.
Good night, Justin. Good night, Kiki and Kai.
Oh, and Kai. Of course.
Good night, everyone. Thank you so much for watching, and do hope that you will return again next week for more This Week in Science. I will try to make it. I don't know if I'll be able to based on circumstances, but the show will go on. Thanks, everybody. Have a wonderful sciencey week. Isn't it like there's like celebration of whales this week, celebration of cats,
Celebration of
dogs, bees. I don't know. Yeah, lots of animal celebrations.
Lion days this weekend. I remember
Lion
that,
days
yeah.
this weekend. That's right. Rawr. And there's something on the 12th that was something I thought was interesting too. Oh yeah, and there's a meteor shower. Get it, everybody. Enjoy.
Yeah.
Science all around. Science all around. Till next time.